Okay, welcome back to the symposium talks. Our next talk is by Dr. Melina Jobbins. She's a postdoctoral fellow in paleobiology at the University of Manitoba's Department of Earth Sciences. So she came here all the way from Winnipeg to have the Winnipeg weather. Okay, sorry about that. Anyway, originally from France, she did her master's in the UK, her PhD in Switzerland, and is now continuing her research here in Canada. Her work focuses on early vertebrates and their central role in reconstructing the evolution of teeth and jaws. In particular, she's a fondness for placoderms, an extinct fish group that are among the first to exhibit both structures.
Thank you, Mona. Thank you for the introduction. Good morning, everyone. Thank you all for being here today. Just in case you didn't have enough fish, we have more of them. So hang in there. As I mentioned, coming all the way from Winnipeg is almost as cold as here, so you guys are doing pretty good. You know, it was pretty nice. It felt like home. Home Winnipeg, not home France. I do miss the weather there sometimes, although it's raining a lot right now. So let's just dive into it and talk about one of my favorite topics ever, placoderms. A bit biased given I've done my PhD on them, but hopefully by the end of that presentation, you guys will at least know about them if you don't know about them yet, and get a bit of a fondness for them too, at least some interest in some of these early fish. And so I just kind of divided up the talk a little bit here, like a bit of a breakdown so everyone follows along. So we'll just start with a bit of background, just to get everyone on the same page. Because, well, I love early vertebrates, but not everyone necessarily works with them. And then move on a bit into the diversity that we have and some of the things I've worked on in the Devonian period, Late Devonian first, and then Middle Devonian after, like at the moment. And then putting all this into a summary and a bit of ongoing work and what we should be looking at in the future and things that projects I'm also being a part of in addition. So, well, I said placoderms quite a few times already, so I'm just going to start with what in the world are placoderms? Well, you have a couple of examples here. Let's just start from the beginning. Placoderms are fish. Here we go. They're some of our oldest, not direct ancestors. We have fish nowadays, right? We've seen some examples. We won't see them. Many of you guys eat them. There's a lot of taxa that comprise like over half of the vertebrate modern groups. Right now, the ones I want to focus on are the early vertebrates. So basically some of these first vertebrates. And these early groups are very important to help us understand the origin, but also the early evolution and to some extent the development as well of some of the very important structures to us, including bone, but our own body plan, how we got limbs, so arms, legs, how did we end up having bone, cartilage, how do we have jaws and teeth? Super important as well. So all these questions stem from looking at these early groups. We can divide early vertebrates into four main groups. So I'm just going to skim through them a little bit and then focus on my favorite. So I'm going to start with your placoderms, which is the oldest one. And here's a bit of the diversity of what they look like on the picture. You can see pretty much characterized typically by this bony armor, this big dermal shield that we have at the front of the fish. There's a little exception here, which I'm not going to go into detail, but overall the characteristics are that they have this dermal shield at the front right there. And they're also very important to understand the origin of the vertebrate body plan. So how did we get to have, in the case of fish, fins, and in our case, limbs? So they're very important for this question. They lived from the Ordovician period right here until the end of the Devonian. So you have the time scale as well to help you a little bit with the ages right there. So we're talking in hundreds of millions of years here. So we're stepping back in time just a little bit. The other group that we have are the Chondrichthyans. We do have, of course, groups still living nowadays. These are the cartilaginous fish, so they include sharks, rays, skates, and ancestors. When we talk about early vertebrates, in the case of Chondrichthyans, I'm kind of restricting it to the times of the Silurian and Devonian-ish type of period. So with this early evolution ongoing in these groups. We also have the Osteichthyans, so the bony fish, which you guys think a lot when we think of fish. Well, there we go. These little guys. They live from as early as the Silurian, same thing as the Chondrichthyans, the cartilaginous fish, and same kind of thing. We think for early vertebrates, in the case of the bony fish, we think of similar time periods until the end of the Devonian, maybe going into the Carboniferous a little bit. In my case, I'm kind of stopping at the end of the Devonian. We can divide bony fish into two main groups. So the actinopterygians, or the ray fin fish, which are characterized by the fins having these like ray, very thin-like structures in the fins. And this basically includes 99% of living bony fish. So if you think of a fish, chances are you're thinking of a ray fin fish. The other group, the lobe finfish, is characterized by musculature in the limbs, in the fins, eventually turning into limbs for us. So that's where we come from. And also this whole transition from water to land also happening. And we can see these structures really developing, the bony structures developing in the fin as well. So they include us, of course, the tetrapods, 4 limbs, but they also include the coelacanth and the lungfish as well. And finally, my favorite group, the placoderms. Placoderms lived from the Silurian period as well, till the end of the Devonian. They were most diverse during the Devonian period. Like many of these early vertebrate groups, there was actually such an abundance of fish groups at that time, evolving and also just diversifying, that we call the Devonian period specifically the age of fishes. And placoderms unfortunately go extinct at the end of that group, at the end of that time, same thing as the ostracoderms. Still questions around why, and that's like an ongoing question. Not too much research has been done about it, but we're looking at that at the moment. Can't say much more as it's early in that research. Placoderms are what we call stem-jawed vertebrates, so basically on that ancestral lineage that leads to the true forms of jawed vertebrates. And the key to understanding the origin again, early evolution, and the development of some of our key structures, including jaws and teeth, but also a few things like certain types of bone, the bone that we have, called endochondral bone, and also looking at something like internal fertilization. The characteristics include a skull and bony armor, as we can see, kind of similar to what you see in the ostracoderm that I showed you. So they have the shield at the very front. You can see the variety of groups we have here, and they all have this shield in the front there. And that's very characteristic of the placoderms. All this is made of bone, dermal bone, while the rest of the skeleton is actually made of cartilage that is only lined with a very thin layer of bone. So this preserves very poorly. So what we tend to find would actually be just the elements of that dermal armor, of that big, thick, bony armor at the front. They are divided, as you can see, into a couple of groups, 6 to 7 orders, depending on who you ask. The phylogenies can vary a little bit, and we'll dive into phylogeny just a tad later on. But the group I focus on particularly is the Arthrodire group. So if you hear Arthrodires, this is what I'm talking about, which is the most common and the most diverse group of placoderms. And they have 2 main defining features for those who are curious. The first one is called a nuchal gap, which is that gap that we see right here on top between the head and the thoracic armor. And the upper jaw is made of 2 elements, which is the anterior and the posterior supragnatals. Now, placoderms have a gift of having different names for every single bone than what we have. So I'm not going to give any names to confuse anyone. We're working on hopefully getting that, synonymized at some point, but that might take some time. So I'll stick to upper jaws and lower jaws, don't you worry. But you have a very good example here of the most famous Arthrodire, most famous placoderm probably, Duncleostus. But there's still a big diversity, as you can see from the image right here on the left. Putting all this in perspective, this is great. I'm showing you all these cool groups, but what do we do with them really? Well, there's a lot of things that we look at and that I like to look into. And I kind of divided that into 3 main topics. So of course, we want to look at the phylogenetic aspect, so understanding their diversity, but also establishing better relationships between these groups, but as well, how are we related to these groups as well? Depending on the kind of fossil that we get and their preservation, we can answer different questions on the more anatomical and behavioral side. So we can look at what kind of diets we have, feeding strategies, also simply what kind of vertebrate structures were already present, try to answer how and when did they evolve, and then also if you have some things that are really nicely preserved with body outline, for example, the body details, maybe we can answer things like swimming habits or swimming strategies as well, which kind of links us to the ecological aspect as well, and helping us understand a bit more about the diversity of the faunas on a more local scale, just like on a global scale, looking at if we get lucky enough and find them in different areas, looking at the migratory patterns, food webs, and of course just looking as well at what else is around and found with it to understand the environment, what we have there and the ecological niches that they fill. I'm not going to discuss all this here, but I'm kind of focusing over my research on some of these main questions I tried to highlight a bit. So the taxon diversity, how it's linked ecologically, and a few other things related to the fossils that I have. And I'm going to start to show you a few things from actually where I did my PhD. Well, actually, I did it in Zurich, but the material I got to work with was in collaboration with people in Morocco. So I was lucky enough to be able to go there 4 times to do field work. We also did this as a field course, so we could take students with us, and we got to show them the sites. They got to learn a bit of geology, how to make a stratigraphic log, and so on. Honestly, it was great. I put a few pictures, I had to, so you guys can also enjoy a little bit and kind of travel back to a different country a bit. First time I went was actually the famous year of 2020, February. So that was a bad idea. But things were great, I at first I told my PhD advisor, I don't know, are you sure we should? Like, I can kind of see there's something spreading around the world. Do we really want to travel right now? I don't know. He's like, yeah, we'll be fine. I'm like, okay, I trust you. Don't. Really don't. But things were great. We spent the first 2 days in the field. I got to find my first little piece of placoderm bone. I was like, yay, I don't know what it is. I just know it's placoderm. Awesome. I'm happy. We got to try some of the local cuisines. So there's some tagine right here. And then, well, that happened. So I thought just a little bit of going back in time, not too much, So that was the lovely airport of Marrakesh when everyone panicked and the Schengen zone started closing. We got incredibly lucky though and managed to leave basically with the last flight before the whole airport closed and the Schengen zone closed. It was a little chaotic and dramatic, but we all made it back. And we got some beautiful fossils to work with and it didn't stop us from going back. So how we do this? Well, we go to the field without taking into account that 2020. We'll rent these beautiful 4x4s, very important to go in the desert. And of course, when I say desert, we don't find fossils here, although we do stop by to take a look at the dunes. Very cool, highly recommend. It's very cool. We're thinking more of something like this. This is the kind of terrain that we work with, that we go into for driving. So we have to be a bit more careful. It's gorgeous though, beautiful mountains all around us. It's the anti-Atlas region, by the way. So we do have to drive through the high Atlas, which is also very impressive views. And of course, as you can imagine, driving through some of these terrains, we're gonna put more landscapes. So we do drive through some of these very bouldery type looking sceneries on that side here. We also do camping, which is awesome. It's a great experience. There's a few spiky things on the ground that you have to kick out of the way, but otherwise worth it. Absolutely great experience. Driving on these kinds of roads, well, sometimes a few accidents happen to the tires. And again, we're in a country where they don't really like to give you spare tires, so sometimes that leaves you stranded, particularly with that middle one here. That second picture, we got stranded for a couple of hours until someone from our hotel came by and was like, hi, you guys went missing. Are you guys okay? It's like, no, but thank you for coming. So instead, well, we took advantage of, waiting to look a bit around and look at what rocks we can find, just basically looking at new areas, see if we find more fossils, and also get some visits from the local fauna. That's pretty cool. Could be worse place to be stranded, honestly. Once we finish looking at all the beautiful camels, we do find some really cool fossil sites. And this is the one that I've been working on a lot, like extensively during my PhD back then, which is called the thylakocephalon layer, which is specifically that layer right here. That actually doesn't outcrop in the picture, but outcrops further out of this picture. And we named it the thylakocephalon layer after these fossils right here, thylakocephalons, which are some tiny little crustaceans. In this case, they're about 5 centimeters big. Why did we name it that way? Well, simply because there's a huge abundance of them. So when that layer outcrops, you can see a picture of it here. You have all these red nodules everywhere on the ground, and almost every one you crack, you'll find one of these crustaceans. So that's really cool. So we don't find the ammonoids in that layer, but we find these little guys. And we also can find really nice preservation in a couple of them. So that's really cool too. We can find, in this case, for example, the eye right here. And then do check out the paper if you want to learn more. I'm not going to go into detail, but we have a few internal structures that were preserved as well once we sent it to a CT scanner. So that was really cool. But also very promising because then we can find other fossils in the area. So we just keep on looking. And, well, we did find more. We found some sharks. We found some placoderms. And now we're looking at the placoderms in this case. So just for context, this is where we are. Well, where Morocco is, excuse me, at the time during that Late Devonian period. So about 370, 365 million years ago. And for those who know, the more famous site from the Late Devonian, the Cleveland Shale, this is where it would be. So just on the other side of the ocean, on the other side of the Great Ocean. So we can do some comparisons possibly with some of the fauna that we find, or at least that's what we were looking for. And I'm going to start with the first one called Amazigthes trinashticae, a reconstruction made by my PhD supervisor, Christian Klug. And trinashticae named after Kate Trinashtik, which is a big placoderm expert from Australia, who's done crazy amounts of work on placoderms throughout her career. So we wanted to acknowledge that in a way. And Amazigthes, which was named after the local Amazigh community in Morocco. So we wanted to give them a bit of a tribute for all of their help and hosting us on their land. So we decided to name the genus after them. And we have some beautiful specimens, as you can see here, also in some kind of almost like a nodule as well. Can you see what's here? It's beautiful, isn't it? It's a nice little blob. Don't worry, I have a line drawing right here. And a couple of little pictures. There we go. just a bit of description to help you guys. I'm kind of used to always showing this fossil, even in person, where you can see a bit more, showing it to my colleagues when I was back in Zurich, and they look at this and I'm like, it's a rock. It's like, yes, it is a rock, but there's way more in there than meets the eye. And we can see, well, the imprint of the skull, of the skull roof, so the top of the skull, the thoracic armor as well. But we also find, which is even cooler, that body outline. So we have a body outline preserved that includes the dorsal fin, includes the pectoral fins. We don't have the pelvic fin, but we do have what we call the pelvic girdle, so the bones of it. So that helps us to do a bit of a reconstruction of what the fin could have looked like anyway. We have, of course, the tail, and we have this really funky structure here, which I'm going to get to in a minute, that we call a lateral keel type of structure. So there's a lot that we can see from that fossil already. And it's really cool because we have so few known body morphologies, like based on fossil evidence in early vertebrates and placoderms in particular. So it's a very cool find. Here's a bit of a reconstruction of the animal. And I was mentioning that little keel right here, which is located in this area. Here's a bit of a zoom on it. For those who may be curious about whether it's preservation bias, we ruled this out because there's a couple of specimens that show it in the same place. So that to us says it's something that's there in the animal. And given the location of it, we compared this with something homologous or similar to what we see in some of the tuna fish or some of the fast swimming sharks, which is a lateral keel. So it basically helps them with swimming efficiency, swimming fast, kind of muscular structure. Possibly based on that, we kind of thinking, okay, we can make some ecological inferences already. We're having a fast swimmer possibly, or at least can make bursts of fast swimming. What does the rest of the body tell us? Well, we look at the shape and compare this to the other very well-known body morphologies of Arthrodires, which is Coccosteus right here from Scotland, and African aspis from South Africa. And there are some key differences here, including the ones I've highlighted with the placement and shape of the dorsal fin, which is much more posterior in all of the known placoderms, actually. Like Amazictes, is the first one to show the placement of that dorsal fin so close to the front, so similar to what we see in some of these fast swimming sharks. So we have a very shark-like looking placoderm right here. So we did suspect, of course, that this would have been present at the time, but we didn't have any fossil evidence for this until Amazictes came up. And of course, the shape of the tail right here, right? Same thing. To add a bit more to that comparative anatomy, we also did a PCA, so we did a specific analysis with a colleague of mine, Humberto Ferron from Spain, who plotted our little guy, the body shape and the tail shape, because the tail shape is one of the most important parts to infer the swimming type, the ecological group of these fish in particular, but especially sharks. And so we compare them. In this case, we compare our little fish to sharks as they're the closest related to them in a way, but that still has descendants nowadays, because then we can actually infer from things we see and that we know. So we got everything basically to modern ones, and this works perfectly well with what we've been saying with our comparative anatomy, which is great. So we have what we call a macropelagic swimmer that's capable of high swimming speeds. Moving on to my little baby. I love him. This is Anacanthus. Anacanthus, I really like this fish because he has some very cool, there's a cool story into it, which is, well, first look at it. It's great. It's a freak. I keep calling it a freak and I love it. But it has a great story because it was initially found actually in Poland. I didn't get to name that one. It was found over half a century ago by a Polish researcher. And that's pretty much all that they found at the time in the Holy Cross Mountains. And he didn't really know quite what to make of it. So suggested it might be something called a fin spine. So in some sharks especially, we can find at the base of the fin, we can find this bony spine that basically comes along on the side of the fin. So we suggested it might be one of these, but in possibly a placoderm, question mark. He had the placoderm right, but as we found more material from Poland, but also actually Morocco with skull roof remains as well, we got to figure out that it's actually the lower jaw. It was not a fin spine. So we actually have just a very weird fish, a very weird placoderm with an incredibly long underbite. And thanks to this specimen in particular that has like almost a complete skull preserved, with a few things displaced there and there. So we managed to reconstruct this nicely. I'm not going to go into details with the names of the plates or anything again, because they're also different. But you can see already there the length of that lower jaw compared to the rest of the skull, right? And this is not even fully complete. So here's a nice reconstruction of it, like a line reconstruction. And when we put all these jaw elements back in place, the upper jaw seems to be pretty much aligned with the rest, maybe a tiny bit forward, but especially that lower jaw that has this huge underbite that we haven't seen before in any of these early groups. So that adds something very new to our knowledge. And of course, raises a whole lot of questions, why, how, etc. And of course, having such a weird lower jaw, there's going to be a whole bunch of other features, other characteristics that are going to go with it, including the upper jaws that are going to be shaped very differently as well. So you can see here the shape of these jaws are much more V-shaped or Y-shaped for the lower one. While what we see typically in other groups, well, other placoderms, is very U-shaped, like very rounded mouth, right? Which is, of course, what we see also in sharks, but even in ourselves. So this is very different in this animal right here, which highlights the uniqueness of the animal. And so we're trying to do a bit of work to understand, well, what it's used for. So we're actually doing we're hoping to get some of the ongoing research getting done maybe within the next year or two, using the synchrotron in a very fancy CT scanner in Grenoble in France. We're waiting to hear back to see if that's going to be possible. But in the meantime, we can actually look at the more external part and see what we can say from it. So we can look at the teeth, for example, and we can notice that the teeth is not the best picture. I'm sorry about that. Points slightly to the back, as well as to the side, as well as lingually, so inside. So it points it has a very odd direction, which we kind of see in some modern animals when we try to look to try and understand what it could be used for. And some examples would be like the bowfin fish or the salmon, which would be a function to be trapping live prey. So we could suggest that this animal had a function similar to this. And then for this long, weird elongation, well, that's the part that we're still looking into, that we're still trying to understand. So there are some ideas, but because the animal is so big, we also need to take that into consideration because the functions can change as well depending on size. What else do we have that has an underbite? Well, we have a couple of them, not that many, especially with like more extreme cases. So this is the oldest case that we have in the record. And we have one example here with a chondrichthyon, so a cartilaginous fish, ornithoprion from the Carboniferous. We have a cetacean from the Pliocene, so much more recent. And on the modern scale, we have something called a half-beak. So this is the most extreme case, as you can see how elongated that jaw is, right? The underbite. So some people would ask, well, can you infer basically by taking what you do with the half-beak than what you do with an anacanthus? Well, it's very tricky because the half-beak is up to 30 centimeters in size, so very small, while the skull of an anacanthus is already bigger than that. So like the smallest set of jaws that we have, which is possibly from a juvenile, were 30 centimeters. So like making inferences from that is much more challenging, especially since observations that were made for the half-beak's use of its underbite would be things like sieving through sediment or going and poking into little holes and crevices to get prey to come out. I can't really see an anacanthus, a big fat fish, doing that really, like poking into holes with its big underbite. So it's still ongoing work. But we did do a bit of phylogeny work, so looking at the relationships between these taxa, and I've highlighted this here into this group. So we have Amazicles right there and an anacanthus within a cluster that's not very well resolved, so we don't know the relationships well within that group, but they belong to what is called the Selenosteids, which is a small little family that is typically characterized by these large eyes and associated characters like a short cheek region and so on. They also have standard jaws with more teeth on them, and anacanthus being kind of the exception with having slightly bigger teeth compared to the other guys. And of course, they all lived from the Late Devonian, and they lived in shallow seas, which works well with what we have here as well, because it was a relatively shallow sea. We're talking a maximum depth of 200 meters. Just a brief mention that we do have, because we find it in Poland and in Morocco, some evidence for migration, which is great, and it's actually also supported by the invertebrate fossil record in the past. So we have a bit more evidence adding to that possibility of migration happening within that Arctic Ocean right here. But it's great. I love the Late Devonian. We did some great stuff, but also what happens a bit early. I want to go back in time a bit more into the Middle Devonian and what I'm doing at the moment for my different postdocs. Here we are, around 390 million years ago or so. What I would be working on is, again, still trying to understand that fish diversification that's happening before Late Devonian, now Middle Devonian, trying to understand a bit more between the different times, what happens, and assessing some, particularly between the Euphilian and Givesian, so these two time periods right here, trying to understand what groups changed, whether we have higher abundance of certain groups versus less of some others, and see if it kind of follows a general trend that's been suggested in the literature. And I'm kind of focusing for this in Manitoba because, well, that's where I am, so I might as well. Might as well see what they have here. And it's been underexplored, so we might as well take advantage of it. We just had this paper published actually just this week, so we get to show it now, in Canadian Journal of Zoology, in which we're showing some of the diversity that we have from this one locality, one formation called the Elm Point Formation, So back at that time in the Middle Devonian, we actually had this huge basin that you can see here that actually also included a lot of Alberta, some of Saskatchewan, and a little bit of Manitoba, just enough for me to be looking at. And we have a couple of quarries alongside that one lake called Lake Manitoba right here. This is great for us because it's actually not easy to find good outcrops without the quarries. In Canada, it's been kind of tricky, especially in like some of the prairies, because we don't have all that, like, especially like Saskatchewan, Manitoba, we don't have all these mountains, right? Or it's much more limited. Having the quarry owners being able to help us with that and giving us access to check out what fossils are found is amazing. Like, we're very happy for that, very thankful to them for that. We got to go out there, go and check out the sites, try to look for some cool fossils. We got to go with Joy Moisyuk, the curator of the Manitoba Museum, and my postdoc advisors, Ricardo Silva and Kirsten Brink right here. And you can actually see Joe and I right there working on extracting a small specimen that Joe found, which is this little guy. I like to say it was an easy extraction, but it was really just a pain in my butt because it was very fragmented and it was a lot of puzzling afterwards. So that took a bit of time to reconstruct. You know what? Worth it anyway, because we get this so little right now in the collections that every specimen basically counts. So we able to add more as we go, as we keep going back to these quarries, especially the ones that are active and ongoing. These are the best ones because we can keep getting more. And well, we find a bit of a variety of things, actually, more diverse than what we expected for the fauna at the time in this region. We thought there was very little things, but we do have a bit more, and it shows promise to keep searching, because this is only from the last 2 years of looking in the summertime, which was not that many times, it was a handful of times. So I think it shows great potential to find more in the future. And of course, the first thing that we find are placoderms, yay! But also a bit more of the other groups too. So we find, of course, the Arthrodire group, which is the most common one, as I've mentioned. And here's a few examples with a skull roof, partial skull roof, of something called L. mastus, which I'll get back to in a minute. For context here is, it's located from above. So we basically have, when I say skull roof, is this area here. And we're looking at the orbit here from the top. So this is where the eye would be. We have the cheek region where that would be. And then we have right here the two insertions for the condyles for the articulation with the thoracic armor. So instead of having one articulation point like we do with the neck, they have two of them, one on the left, one on the right. So the lateral movement is a bit more limited, of course, but they do have much more abilities for muscle then build up, muscle strength, have much more muscle strength for the up and down motion, which I can imagine is because, well, this is much heavier. The dermal armor is usually pretty thick in these groups. So that is pretty helpful for them, and also it helps them with power with jaw opening and closing. And speaking of jaw, well, we have a cool jaw right here from something called Squamatoognathus from Steep Rock, which is a partial one, but hopefully we get to find more eventually. We've been going to the quarries where that was found, but infilled with water, so sadly that's something to see what we can do about in the future. Hopefully there'll be a solution that we can figure out with the RM. But we also find other placoderms, some of them called the Placoderms, which are some bottom-dwelling fish, so living closer to the sea floor kind of animals. And they had more reduced bony armor, so they still had some, but you can see less of it. So that is actually hard to find because they're much less well fused together. So what we tend to find in these groups are just the jaw elements. And here's a couple of examples here. This is seen from above, so it's a lower jaw, and from the side. And then you can see some of the jaws upper and lower, of another kind of animal, Rhynchodus, two Placoderms, and both of them being shell-crushing animals, as you can see, especially with Rhynchodus here, right, with that big crushing plate. We also find bony fish, though, which is cool, including from one extinct group, the Onychodont, and we would find especially teeth right here from this region called the symphysial area, from a tooth roll. So basically the jaws come together, and in this region they have this row of teeth that just comes out like this. So we find a few of these teeth, and we also find a couple of scale pieces or pieces of head bone that have odontos, so tail-like little tubercles on them for ornamentation. So you can see an example right here of some, and you can see that on hoof-shaped tubercle that we have all over as well. We also find Sarcopterygians, so the lobe-fin fish, so the ones that we come from. We have some lungfish tooth plates in particular that we find a few of them of, which are pretty nice. Some can be very well preserved, such as this one, and some a bit less well preserved, like this one here, which makes it a bit harder for the ID at the genus level. Unfortunately, it wasn't possible, but we can still identify that as lungfish though, so that's great. And we have something which is actually for everyone the most exciting find, which to some people may look like the most boring because it just looks like a random piece of bone. which is the one right here, this square-shaped one almost, which is from something called an osteolapiform, so on the tetrapodomorph lineage. So basically, we are looking at the fish stage of that water to land transition, the very, very beginning of that water to land transition. When we still fish, we find the oldest remains in Canada right now of this, with that little bone right here. So we're really hoping we can find more of that with time as we keep going, keep exploring, keep chatting with the people there. Fingers crossed. So we compare this a bit with other North American faunas to see what we have. So our formation is in the aphelion time, so that first stage in the Middle Devonian, and we compared that with a few other ones known from the US, and as well with some of them known from the Juvesian time, so we can see that second stage, so we can see what changes over time. Well, the first conclusion that we can say, this is the numbers are per specimen, so it looks pretty sad. So that's the first conclusion. It is incredibly undersampled and understudied. And actually, that's really the big conclusion that we have. There needs to be more work done on all of these faunas, all of these groups, all of these places, so we can really understand what's happening in that Middle Devonian period with the diversification of these fishes. But looking at the Arthrodires in particular, there may be, again, all this has to be taken with a grain of salt, right? Because it's so undersampled. But if we're looking at some of the ones with the best sampling, so we're looking, for example, at the Elm Point in the early stage, and then in the later stage, we can look at the Milwaukee Formation. We may have a bit more of an increase of these Arthrodires, which is kind of what we're supposed to be seeing in the general trend. So that would be something to further explore and see if we can confirm that trend once we get more samples, more studies going on with these different places. But the one I want to focus on now the most is Elm Osteus, which was published just last year, and I was lucky enough to actually get decent media coverage as well, especially in Manitoba and some of Saskatchewan too, which was amazing. We nicknamed it Elmo, so you guys can call it Elmo if you want. It was published for the first time back in '96, actually, also by the U of M, by some researchers at the U of M, a student in particular, Gavin Henke. It was attributed to another genus that was existing already called Eastmanosteus. Eastmanosteus was often seen, you can see the name here in the title, was kind of considered, without never being discussed, as something called a wastebasket, which is basically what we can see here as an example of a wastebasket. Let's say you take taxon A, D, and G. No way are they related, as in like forming one cluster, one group. That's what we call a wastebasket. So we take a whole bunch of different things, toss them into one, and say, yep, that's our genus. So we kind of suspected it, and we're pretty certain that that was the case for the Eastmanosteus. So we took advantage of looking and revising this animal so that we can look at the phylogenetics a little bit of that group so that we can show what we have and draw conclusions from that. We got to rework on Elm Osteus, well, the now named Elm Osteus, spoiler, I guess. Here's a new drawing reconstruction, one of the skull roots, the holotype actually of the animal. So again, you see where the orbits would be right here, and then the back of the head right here. And it's a nice little reconstruction here. So this is where the orbit would be. And then you have the articulations here to go with the thoracic armor. We compared this to the most and best known Eastmanosteus other species. So two of them from the US, Pistolosis and Magnificus, and another one from Australia called Eastmanosteus calliaspis. So you can actually, I'm not going to go into detail with the descriptions of them and the discussion because it's very finicky and very detailed, but you can already kind of see some differences, right? Especially in the shape, for example, the overall shape of the skull, and also in the details are also different. So we put this into a character matrix, which is basically what gives us our phylogeny. And well, the results is Eastmanosteus is kind of all over the place. It doesn't cluster in a way that we want, as in monophyletic. So it doesn't come in that way, right? We have one here, two of them there, not together. And the second analysis that we run, parsimony, thanks to Joe Moisiuk, he did the analysis for both of them with us. And same thing, same results. Eastmanosteus is kind of all over, doesn't cluster as a monophyletic group. So it is indeed what we call polyphyletic or a wastebasket. We also did a Bayesian probability test, which is a very fancy little statistical test that Joe did, which you basically ask the software with the phylogenies that you get, what are the chances of these four groups being the same genus? Well, the results are pretty low, 0.004. So I think we can safely assume that this is different. So that's why we renamed the Manitoban taxon Elm Osteus. And that also means that there'll be research that needs to be done, revisions that needs to be done on these other species as well to better understand them, see where they plot again, and then give them possibly new names. So to put this all together for a little summary, because I've been babbling about placoderms for too long now. Thank you for following. So we have, on the phylogenetic side, we have this rich diversity of Arthrodires and also other vertebrates that lived in the Middle to Late Devonian. We have a bit of an increase in the resolution in the phylogeny, which I didn't discuss really. That's because there's still a lot of work that needs to be done to clarify some of the relationships, especially within the groups. And a lot of that will be working on the character matrix. We have, on the anatomical aspect, thanks to some really nicely preserved specimens, we can describe new body morphology, some new features that were not even described before, including that lateral-like keel. We have the oldest case of lower jaw elongation, which overall increases our knowledge of the diversity that we have of Arthrodires and placoderms. And then on the ecological aspect, we have evidence for the migration I've mentioned across the Arctic Ocean, thanks to Eniacanthus, evidence for high swimming speeds in placoderms, thanks to the fossil record now with Amazicles, and some very odd feeding strategies that we haven't seen before, possibly with Eniacanthus again, which all this brings to this diverse fauna that we have in these different areas. And for future work instead, I'm going to go with that. Improving on the placoderm character matrix, so improving on that phylogeny, is really key to help us not just understand the group itself, but understand the relationships regarding us as well related to it, and how all these different features that we have evolved through time, starting with these very weird groups. Of course, connected to that, we need to revise the past work on also these North American, in this case, Devonian vertebrates, and worldwide, of course, is always useful, which will lead us, of course, to full-on reconstructions for all the vertebrate sites, so we get to work on more environmental and ecological aspects of it too, which will then hopefully give us a nice updated version of that early vertebrate and placoderm diversification through time that we could then use in other research. And well, to finish up with what we're doing right now, well, the right now, because it's submitted, I can't tell you, but I can show you what we're doing with, hopefully with a student this summer, which will be looking at L-mostinx actually again. Now that we've published on it, we, surprise, we just donated a specimen that's even more complete than what we had. So now we can redescribe it again. So that will be a job for a student to be describing in particular all of that thoracic part here, because only one bone of it was known before. So now we can describe a lot more of it and focus a bit on this region here, which has little denticles in there, right, with a fin insert. So that's a pretty neat thing happening. So we're going to look a bit into that. The objective with this, in addition to the research, would be to have a full reconstruction of the placoderm that we can use for exhibit purposes. So kind of like what we see with Dunkleosteus across different museums, we like to do that with one of the local ones with L-mosteus and eventually have that in our museums. Which, speaking of, if you ever pop by the Winnipeg, stop by the University of Manitoba, we have a small exhibit on L-mosteus and placoderms right now. We're hoping it's going to be there for at least a few years. If any of you happen to just drop by Winnipeg, which I know it's low, but don't hesitate. And the other one that we want to do is looking at the Middle Devonian again, so starting a bit understanding the Middle Devonian sites a bit more. So we're going to look into the Middle Devonian of Ontario with two different formations. When we have a lot of actually micros that we have. You're doing me happy. There's some really cool stuff we're hoping to identify. This is only a small handful of them. There's a few thousands of specimens just collected by this one guy, Bob. And that's amazing. He does a great job. And I'm hoping to get some good collaborations going with him thanks to his hard work with sampling over the years. And one other project I'm really excited about, which is these right here, which are jaws from an Arthrodire as well, but have these very weird possible teeth. We're waiting on the results of the CT to check the structure inside to confirm that they are teeth. But if they are, they're basically scattered all over the jaw instead of being like this one row. So this is very exciting to try and understand what happened with this guy. So somehow I keep finding the weird stuff. That's great. I'm happy. That will be hopefully the next project to come out within the next year or so. And with all that, thank you all for listening. Thank you for tolerating fish for so long. And thank you, Mona and Matt and Eric and the rest of the APS for a great conference. Thank you.
We do have time for some questions.
Damn
it. I have a question.
Okay.
Sorry.
All the Arthrodires,
Yes.
Dunkleosteus, right?
Yeah.
They have these plates in front of their jaws.
Yeah, so these would be the jaws, basically.
Yeah, I think on your slide you call it similar to dentine or kind of an early form of dentine.
Yeah, so there are some animals. So placoderms have something called semi-dentine, which is a very specific type of dentine, like derived from dentine. We don't understand fully the relationships between dentine and semi-dentine, but there is understanding that there's a correlation between them. And most of the jaw is made of some of that, yeah.
Do you think these plates came in only once during the life of the fish? So that small fish would have small versions as the fish got bigger, they just got taller, and they didn't have any roots on these plates, right? These teeth plates.
That's right, yeah. So
there must have been some mechanism for these teeth to grow.
The teeth of placoderms is very, I didn't go into detail, it's very intriguing. There's actually some debate about, for some people, not wanting to call them teeth, because despite them having a pulp cavity, especially early on, it actually gets infilled with time. So we can see changes happening as they get older, to accommodate for the teeth basically getting worn down as they get older, which could also lead to changes in diets, which can happen. Like actually some juvenile Dunkleosteus had some teeth to start with.
So potentially
and change to blade. Yeah, so they basically just go once as they're young and then it gets worn down. So it actually makes it also trickier for ontogeny sometimes, to see is it a juvenile versus a small adult, because we need to find the young version sometimes to confirm these things, because they can change quite a bit. So they'll have one shape when they're young, but as it can change significantly as it grows older, because it grows in different directions. So it doesn't just grow anteriorly, it goes also posteriorly, it grows ventrally, it grows in different directions, which can change the whole shape of the jaw. It's a little messy.
Yeah, potentially these fish would have swallowed smaller fish whole instead of trying to take a bite out of it.
A few of them would have probably done something like that, yeah, especially the trapping life prey kind of ones, like in Acanthus, wouldn't be surprised. It would also not be surprising that they would prey on usually more juvenile ones, because the bone would be more fragile, it would be much thinner, and it would thicken as well with time for some of them at least. We don't have the ontogeny for all of them, so I can't say for everyone. But the preying on the juveniles is much easier. And then of course, because the whole part behind the shoulder is not made of dermal bone, that would also be a great point of attack for a lot of these predatorial ones.
Okay, thanks. And one other small question. I think I've read that earlier placoderms were mostly freshwater, and later in the Devonian they became more marine. Do you agree with that?
So I do remember that. So I have read that this was the general trend. However, I can't tell that for certain because I haven't searched and I haven't looked into anything that lived from the freshwater. I always worked with the marine kind, so I'm not entirely sure how. Yeah, it's also marine at the time. It's a shallow marine environment as well. So a lot of them were found in shallow marine. There's of course different ones too, but yeah, like they definitely. I do know that they occupied lots of the different environments, including freshwater. But I'm not sure about the trends that they occupy in that sense throughout the time. I'll have to double check that.
Yeah, like we had a field trip last summer. Yeah, that's freshwater. Maybe a little mix of.
Okay, from the yeah. Okay, I have one example for the workshop tomorrow actually. Okay. If you're attending.
Thank you.
What's the current consensus on placoderm monophyletic?
So placoderm being monophyletic, there's a bit of a mix. So there's some research that supports for monophily, so all forming one cluster. But there's also evidence, and I think it's kind of leaning towards that now, being paraphyletic instead. So not including, there is more evidence supporting right now. Or at least I'd say the consensus is going slowly towards paraphyletic more than monophyletic.
The underbite modern fish is tiny. So what's its habit? I mean, I think I'm loud enough. I probably don't need this.
It's for the Zoom.
Well, anyway, so I'm curious. You had one analogy to a modern fish with a severe underbite.
Yeah.
And I'm just curious, why is that? It's not sexual dimorphism. It's a feeding habit.
It's a feeding habit.
How does it feed?
So it's a feeding habit, and there is some of the research is mostly observations that were made for the half-beak, and it was either sieving through sediment. Some people have seen that, and other ones I've seen it looking at just poking into crevices to try and get out some like the small fish, to like basically scare them out. It's through other crevices, and then they trap them that way.
So
Like
things, or that's just a guess of what it does.
So that's all based on observation.
Ah.
Yeah. So there's no paper writing about it in itself, but there are reports instead of observations happening.
We have an Ordovician and a Devonian site over just outside of Cranbrook. Were these fish common enough that we should be keeping an eye out for samples?
Possibly, yeah. Because I'm thinking it's probably within that basin, right? From that outcome
Yeah,
basin.
it's up the Bull River area there.
Yeah. So
And,
it's
but we find lots of marine
okay. I mean, if you
items, but we don't find.
If,
I haven't seen any fish yet.
if you find marine stuff, you could probably find them. So the thing to look out for, especially in that basin, the currents were relatively active, so it was not a calm water because it was more shallow. So most of what you're going to find might actually end up being fragments. So you may not be looking for something that's a full complete specimen, but looking at maybe smaller stuff. But it is likely that you can find something up there, yeah. Ordovician is tricky because I'm not sure the age of the Ordovician. Do you know?
I don't know.
Okay,
I'd have
it's
to
like
buy it in front of me here and
because so far so if you have something from early Ordovician, it's probably not. It's going to be pretty unlikely. Because right now the fossil record doesn't mean you shouldn't look, but like the fossil record right now starts around the Middle Ordovician, and we have more abundance in that late Ordovician time. So you have something from the late Ordovician, especially, it could be worth keeping an eye out for it. And it would be mostly just micro scales.
I have a sample. I'll show you a photo later. Similar to it's a little bigger than holding something in my hand. And I can't tell what it is, and it doesn't look like it's coral or anything, but it's very similar to what you had up there as a picture. So,
okay, sounds great.
okay.
Happy to do it.
In regards to the Alenacanthus, and the half-beak, does a half-beak, I don't know if maybe you said this, but does a half-beak also exhibit the like the lingual posterior facing of teeth? Because I noticed on the salmon you had that, and that was like it showed the male salmon in the breeding form of the face. Is that same teeth style also in the
So we don't see that in the half-beak. I'm trying to remember right now. I'm trying to remember if it even has teeth. If it does, I think it's incredibly small ones. So it would be a very different function to that than what we see.
So could that possibly point to a difference in niche and foraging?
Absolutely, yeah.
Okay, yeah, because like I was kind of wondering if it's got the same teeth style and exaggerated skull features as a salmon, like what do you think might be the possibility that could be like a breeding change, like what is seen in the salmon that also have exaggerated jaws and similar teeth.
So that would probably be something that could be observed with a half-beak. I know that there's still a lot of, it doesn't seem like they did much observations with the half-beak right now, so there could be more, I'm sure, to be done with it. With Alenacanthus, of course, it's much harder to say, right? Because it's something extinct and there's very few specimens. So we can't make any inferences about that. Even sexual dimorphism is challenging. We can't do it at that point because there's too few of them. But it seems to be something repetitive so far from what we have.
Okay, cool. Thank you.
Thank you.
I'm just wondering how was the inference about migration of Alenacanthus was made?
The inference?
Yeah.
Yeah. So it's basically just because we have, let's see if I can pull up, sorry. That's going to be far back. Here we are. And we know where both places are located, right? So we have Poland on one end with one star, and then we have the same on the other. So that's basically what we use as the assumption that we have a migration going on, because we see the same species in both. So they have to be able to be in both areas. That's what we use in the fossil record, often suggesting that there is migration happening. But it's also just useful to say that simply because there is invertebrate fossil record that argues for that too. So by itself, there's more like, it could be living in two different spots, but it's more like we at least know that it's not an endemic thing. So it's not living in one specific area. That would kind of be putting all of that together.
It cannot be migration, but we know.
Yeah, it would be,
population.
It's more like when we talk about the migration there, it's more like it's capable of moving between the areas rather than seasonality or something. Yeah.
I didn't know the idea about placoderms moving from freshwater to marine, but I recall 50 years ago or more that there was an idea that vertebrates, in fact, started off in near shore or non-marine and then moved to the marine. Are both those ideas out the window, or where are they? I'm just curious whether the placoderm story came from the older vertebrate story.
So the oldest record that we have for the oldest vertebrates, we find them in these shallow marine, like very shallow ones.
Like in Quebec and go to Zodium.
So we have some in the oldest ones would actually be from the sandstone formation in the stairway sandstone actually in Australia, and that would be like a coastal kind of environment.
Yeah, the familia was a type of flat type that had these little guys in it.
Yep.
You might want to. I guess when you were looking for microvertebrates, that's how they were found. They were just a little ornamentation on their skin, and they're pretty unique. And so I was checking what I called it out, folks, and that was we found them.
We'd like to thank you for excellent presentation. I'm sure that your EPS membership, as well as she selected our T-shirt. Which unfortunately, well, I guess, let's see, Corey, what else? It has some kind of green stuff on it, right? Oh, Corey's not here. Okay, anyway.
Thank you.
Thank you very much.